Our next presentation, as I said, we're going to be changing our schedule a little bit, will be Dr. Charles Gersbach. Dr. Gersbach is a Rooney Family Associate Professor of Biomedical Engineering at Duke University. Thank you.
Thank you very much for the opportunity to tell you guys about some of our work that we're doing at Duke in the area of epigenome editing. And thanks, I want to echo everyone else's sentiments about the appreciation that we have for Paul Allen and the Frontiers Group for funding this work that's definitely taking us to new and interesting areas that we wouldn't be pursuing otherwise. So my background is in engineering. I'm in an engineering department in a school of engineering with engineering students, but we're working in epigenetics. And so really our goal and the way we think about this is how do we transform epigenetics into something that's really an engineering discipline? And when we think about what defines an engineering discipline, we want to be able to have parts that we can put together and take apart very precisely and easily. And we want to be able to predict how those parts interact together when we recombine them and move them around. And I think arguably we really haven't had those tools or that type of technology for manipulating the epigenome. And so we've been working on developing those tools and then also on a variety of applications of how we would use those as we go and develop them. And so I'm going to maybe highlight a couple of the tools that we've developed and then talk about some of the applications that we're using them in areas like disease modeling, cell programming, and gene therapy. And this has already been introduced earlier in the meeting, so I won't spend too much time on this, but as many of you know, the simplest DNA targeting system that we have right now is the CRISPR-Cas9 system. And while many of you are familiar with Cas9 as the nuclease that cuts DNA for genome editing, you can actually inactivate the cutting function of Cas9 by 2-point mutations to the protein. Well, it will still bind to its guide RNA. It'll still go find its target site in the genome, but it no longer cuts DNA. And so now it becomes this type of modular protein scaffold with which you can attach or recruit different epigenetic modulators to manipulate nearby epigenetic states. And so we and many others in this field, we're all kind of pursuing the development of this form of a dead or nuclease inactive Cas9 for epigenetic editing and gene regulation. And alongside many other groups, one of the first things that we did was attach the VP64 activation domain to this dCas9 protein to create a synthetic transcription factor that we could target to different promoters or regulatory elements and activate gene expression. And when we characterized this very early in the state of this field, we showed that we could take one or more guide RNAs, target it to a gene promoter, and activate the expression of a silent promoter in cells that normally would not be expressing that gene. But a couple of the important observations that we made about this type of gene reactivation in these cells were first the specificity with which we saw gene activation. So by RNA-seq, we saw that we could take a cocktail of guide RNAs or a single guide RNA, target it to a gene promoter, in this case, the promoter of this IL-1RN gene, and show that across the entire transcriptome, the only transcripts that were upregulated by this treatment were the 4 isoforms of IL-1RN. And so this got us excited that this was a very specific tool for going in and doing gain-of-function assays in the human genome. But we also started to think more about how was it that we were actually being able to reactivate these genes that would normally be repressed in these cells. And for that, we wanted to understand, well, what are all the parts that we need to turn the gene on? And it turns out, yes, you need the dCas9 to do the DNA targeting or DNA binding. You need the VP64 in order to do the transactivation. And you need the guide RNA in order to do the targeting to the correct sequence. But we also went and, with Greg Crawford at Duke, we looked at what the starting state was in these cells and saw that by DNA-seq that there's really no open accessible chromatin, at least no strong signal at this promoter in this HEC293 cells where we started doing these experiments where the guide RNA target sites are binding. However, after we deliver the guide RNAs either with dCas9 or dCas9 attached to the activator domain, in both cases we create a weak hypersensitive site at that promoter that is a modest opening of chromatin, but still reproducible and robust. And so this convinced us that you could use this type of synthetic transcriptional activator to go into closed regions of the genome and awaken genes that normally would be repressed in these cells. And so then we started thinking about, well, what are some of the different applications that we could use this for? And of course, we thought about cell reprogramming. And this is this Waddington landscape has been shown in a couple of other presentations in the symposium, where this marble at the top of the hill is the totipotent cell, and as it comes down the landscape, it's differentiating and falling into these valleys in between the hills that are the epigenetic barriers that prevent one cell type from spontaneously deciding to change its epigenetic state and become another cell type. Now, I think traditionally this had been thought of kind of a irreversible path going down this epigenetic landscape, but we know from the reprogramming field that in fact, particularly with overexpression of master transcription factors, that you can in fact either traverse all the way back up to the landscape from taking a differentiated cell to a pluripotent cell, or directly traverse one of these epigenetic barriers and convert one cell type into another. But the typical protocols for doing this involve overexpression of master transcription factors that bind to tens of thousands of sites across the genome and modify genes in the genome in many different ways. But now with these types of DCAS9-based activators, we've had the tools to be able to go in and determine what exactly is necessary or sufficient at very specific sites in the epigenome to reprogram one cell type into another. And so we've explored using these types of tools to understand the epigenetic determinants of cell fate specification for a variety of different cell types. And one of the earliest that we worked on was a conversion of cells into a neuronal fate. And so this built off the work that Marius Werning had done, showing that overexpression of these three master neuronal factors, that he calls the BAM factors for short, could convert either mouse or human fibroblasts into neuronal-like cells. Now, he has also shown that these factors, when overexpressed, go to tens of thousands of sites across the genome and do many different things. And traditionally with these types of reprogramming protocols, when you overexpress them at super high levels, they even go to other sites across the genome that they wouldn't normally belong at when expressed at normal levels in the corresponding cell type. So we asked the question, rather than overexpressing these factors, what if we go in and turn on the endogenous copy of those genes in the genome? And we wanted to do so with a transient transfection of the DCAS9 activator, so that we could go in, the sites would set up at those silent promoters, reactivate the expression of these neuronal, or activate the expression of these neuronal genes. But because it's a transient plasmid transfection, after a couple of days, the DCAS9 activator should go away. And then the question would be, does this stay in a kind of autonomous new neuronal-like phenotype by activating those endogenous master factors? And so in these experiments, we compared either transfection of control plasmid into mouse embryonic fibroblasts or transfection of plasmid that encodes those BAM cDNAs. So this would be the traditional reprogramming method where you're overexpressing the transcription factors, or using the CRISPR activators to activate the expression of the corresponding copy of these genes from its natural position in the chromosomal DNA. And so if we look at expression of each of these factors, when we just look at total mRNA expression, we see none of these are expressed in the control-treated fibroblasts. If we overexpress them from plasmid DNA, then we get very high levels of expression like you might expect. But when we activate them with the CRISPR, it's good news that we can activate the expression of all three of these different factors in these fibroblasts. But of course, we get significantly less, and note the log scale here, significantly lower levels of expression of these factors when we activate them from a single copy or two copies in the genome compared to when we overexpress them from plasmid DNA. But that's when we look at total expression. If we then use primers that only recognize the mRNA transcripts that come from the copy in the genome, then what you see is everything in red on the left gets copied over to the bar graph on the right, because the only place that CRISPR can activate the expression of these genes is from the genomic chromosomal copy. But if the blue bars are dramatically shorter over on the right, which tells us that even though we get very high levels of overexpression of these factors from the transfected plasmid DNA, they're doing a very poor job of activating the corresponding copy in the genome. So that would lead us to think that after a couple of days, when the plasmids are degraded or diluted and the exogenous BAM factors or CRISPR activators go away, that we might get a more stable induction of this new phenotype with the CRISPR activators, because that corresponding copy in the genome has been reprogrammed and activated. And in fact, that's what we see. So if we look a couple of weeks after the plasmid transfection, we see about a doubling of neuronal-like cells with neuronal morphologies expressing pan-neuronal markers and electrophysiological properties of immature neurons. And then to go in and prove to ourselves that this has to do with changing the epigenetic state, those corresponding genomic promoters, we did chip qPCR only 3 days after plasmid transfection for these different active marks that, and in this case, we're just looking at the promoter for ASCL1, although we did it for the other genes as well. So this is the promoter over on the right-hand side, and these are where our guide RNAs are binding. And these are marks that are on in the yellow tracks, and those are correspond to mouse embryonic brain tissue, and they're off in the fibroblast tracks, and those are in blue. And we're looking at both H3K27 acetylation, H3K4 trimethyl, marks of active gene expression, and this is a gene that's on in the developing brain and off in fibroblasts. And what we see is that only when we activate this promoter with the CRISPR activators do we see these activating marks get enriched at that copy in the genome. And so this kind of supports the idea that using the CRISPR activators to go in and change specific epigenetic nodes in these gene networks is maybe a more deterministic and precise way to induce these new cell phenotypes compared to overexpression of master transcription factors that do many other things across the genome. So we're continuing to use this for disease modeling and developing these in vitro cell cultures, and I'll talk maybe a little bit more about that at the end, but we also want to use these in vivo for a variety of different applications. And so we've had to go in and re-engineer some of these tools so that they're compatible with your typical in vivo gene delivery vectors like AAV. And I don't have time to get into the details of the constructs that we developed, but as one proof of principle example, we moved to CRISPR repressors, so the DCAS9 crab fusions, and as a model target focused on the PCSK9 gene that is a popular drug target for when inhibited or repressed, lowers cholesterol levels in the blood. So for that reason, there's been a variety of like antibody-based drugs that have been developed, but those drugs have to be taken continuously to inactivate the PCSK9 protein. We wanted to know, could we one time go in with an epigenetic repressor and suppress the expression of this gene and have a permanent or long-term effect. And in fact, so we encode these repressors with guide RNAs targeted to the PCSK9 promoter on AAV vectors. We deliver them. This is showing repression of PCSK9 levels in the serum out to 6 weeks, but we've actually taken these mice out to 6 months and seen stable repression of PCSK9 and also a corresponding decrease in cholesterol levels in the serum as well. And the last thing I'll mention of where we're taking this, in addition to exploring a variety of other ways that we can use those tools in vivo for gene therapy type applications or for understanding the relationship between epigenetics and animal behavior, we're also trying to use these tools to better understand the non-coding genome. And we've had a couple of talks over the last 2 days talking about non-coding genetic variation and its role in complex disease, and the lack of tools that are available for in a really high throughput and robust way to go in and annotate the function of those different non-coding regions. And so for that reason, we've gone and characterized these epigenome editing tools for being able to both activate and repress these distal gene regulatory elements. And now we are adopting the high throughput CRISPR screening approaches developed by Feng and others, where we make guide RNA libraries targeted to large sets of putative gene regulatory elements and then deliver them into cells that have been either engineered with DCAS9 activators or repressors, select for changes in gene expression or cell phenotype, and then look at those map the selected guide RNAs back to the genome so that we can start to annotate the function of some of these non-coding regions. And we've done that now. We published this last year for mapping different regulatory elements that correspond to a single gene, and now we're expanding on that to map regulatory elements that correspond to particular cell phenotypes or that control many different genes within a locus, such as different hits from GWAS type analyses. So to summarize, we've been working on developing these epigenome editing tools for both gain and loss of function. We've published a variety of studies demonstrating the very high level of specificity. We think that one of the really useful applications of this type of technology will be programming cell phenotypes, and we think that this might allow us to get more mature cell types, maybe even addressing some of these issues of epigenetic age in culture that have been talked about over the last day or two, and eventually maybe these types of cells could be used for, CRISPR-engineered cells could be used for cell therapy. I just mentioned that we want to use those for going in and annotating non-coding regions, and then where we're really excited to move forward with the support of the Allen Foundation funds is to use this in vivo and mouse models to try and better understand the relationships between epigenetics and animal behavior with very, to determine causal relationships using these types of epigenome editing tools, and we're doing that with Ann West in neurobiology at Duke. And so with that, thank you all for sticking around and for your attention, and maybe there's time for a question.
Do we have time for a question or two?
Are there ever instances where you can just recruit the DCAS9 to allow some increased availability, and then you can get endogenous activation of the locus without recruiting, without having the... Sorry, the question was if you could get endogenous activation, whatever that means, of a locus just by recruiting the DCAS9 rather than having the VP16.
Yeah, so if you were able to open up a locus where there were no pioneer factors already being expressed in the cell, and then you open it up and then you make it accessible to them. I don't know that we have a great example of that. I mean, I think that's kind of why I show the slide that shows that it does seem to be that DCAS9 can get into heterochromatin and open it up. And so, but in that case, there was no gene activation. So it was I think that I like that example because it seems to be a good case where chromatin accessibility and transcriptional activation were decoupled from each other. But I think it would be interesting to try an example where you know the corresponding factors are there, but the chromatins were close.
Right, so exactly, I was wondering, you could actually test whether or not it's actually the epigenetic repression of the locus or the absence of a transcription factor that's keeping a gene from being off.
Yeah, right. No, it's a good question.